When reviewing a gene list obtained from statistical analysis, Subio Platform allows you to right-click a gene of interest and quickly open registered web links. In particular, searching GEO Profiles allows you to check in which cells or treatments the expression of that gene increases or decreases in other research datasets. You can also create your own search links. With the integration of AI into Google Search, gene searches have become easier to use than before. For example, let's search for a gene name on Google. Because gene names can sometimes be difficult to distinguish from general terms, it is useful to add gene human. From the URL of the search results page, copy the part up to the search term and paste it into the Weblink editor. Replace the gene name part with the column name gene symbol. Now, a new Google option appears in the right-click menu. You can search directly from any gene. This is not limited to searching gene names. For example, a query such as pathways or networks including gene name is also useful. As before, copy the URL that includes the search query and open the Weblink editor. Click the Add button and register a new link name. After pasting the URL, replace the gene name part with symbol. If you are studying a specific disease, it is also useful to prepare a search phrase such as, "Does gene name relate to disease name?" Search it once on Google, and copy the URL that includes the search keywords. As before, paste the URL and replace the gene name with symbol. Now you can search easily from the right-click menu. With this setup, you no longer need to type search terms into Google every time. You can call up predefined search phrases from the right-click menu and search for any gene. Because the search phrases can be created freely, this method can be applied in many different ways. Next, we will perform enrichment analysis on the gene list extracted by statistical analysis using DAVID Functional Annotation. In the Annotations tab, click the Copy button and copy only the gene ID column. Open the DAVID Functional Annotation page and click the Start Analysis button. Click the Upload User List button and paste the gene ID list. Enter a list name and select the species. Once again, return to Subio Platform and select another gene list. As before, select only the gene ID column and copy it. Using the same procedure, click Upload User List, paste the gene ID list, and set the list name and species. Being able to register multiple lists like this is one of the convenient features of DAVID. To run enrichment analysis, select the gene list to analyze and click the Use button. Then click Functional Annotation tool on the right. Click the Functional Annotation Chart button. The enrichment analysis results are shown in a table for various gene sets, such as GEO terms and pathways. The search box is very useful here. You can filter the results by keyword. For GEO terms, CC, MF, and BP are useful, and filtering by KEGG_pathway is also convenient. You can easily return to the previous page using the tab. Click the List button to show the uploaded lists, then select the down-regulated gene list and click the Use button. Opening the Functional Annotation chart shows the analysis results for the down-regulated genes. For the up-regulated genes, terms such as plasma membrane were found, whereas for the down-regulated genes, nucleus stands out. BP and MF also show large differences between the up-regulated and down-regulated gene groups. Because the results are opened in separate browser tabs, you can switch between tabs and compare them. Filter by KEGG_pathway and check the list of enriched pathways. For KEGG pathway results, clicking the name opens the pathway diagram. It also shows where the uploaded genes are located on the pathway. DAVID allows you to download the results in various file formats. Here, we save them in Excel format. Switch to the up-regulated gene list and download this result in Excel format as well. Another convenient point is that the downloaded file names are easy to understand. Open the Sample Info tab in Subio Platform and drag and drop the downloaded files into the Files field to save them. Attached files can be opened by double-clicking. Because Excel files support filtering, they are useful for narrowing down results by category. Next, let's look at an applied example using ChatGPT. As shown in the differential expression analysis chapter, the gene lists down-regulated by the three RNA-Seqs were quite different from each other. Therefore, we extracted gene lists specifically down-regulated by each RNA, as well as a gene list shared by two or more RNA-Seqs. Upload these gene lists to DAVID and download each enrichment analysis result in Excel format. In addition, for the gene list commonly down-regulated by two or more of the three RNA treatments, download the result in the same way. We then give these 4 Excel files to ChatGPT and ask it to find common items. As a result, keywords that may provide clues linking the individual lists and the shared list were found. Using these keywords, we create corresponding gene lists. In this way, we searched using 6 keywords and created a gene list for each keyword. In the Venn diagram tool of Subio Platform, the Hashtag Overlaps tab allows you to combine and analyze 3 or more gene lists. No genes were shared by all 6 lists. However, 7 genes were shared by 4 or more lists, and 63 genes were shared by 3 or more lists. Here, we create a gene list of the 63 genes shared by 3 or more keywords. When this gene list is compared with the gene list down-regulated by 2 or more of the 3 RNA-Seqs, 1 gene is found. For this gene, we use the Weblink created earlier to search for pathways or networks that include this gene. We also search whether this gene has any known relationship with ESRRA. Furthermore, we check the GO cellular component of this gene and examine its relationship with nucleus. This type of exploration can expand according to the analyst's own ideas.